Hello, Assalamu alaikum. This is me, Alina Batul. Today I will discuss glycol cleavage and glycolysis. Introduction to glycolysis and glycol cleavage. Glycolysis is a fundamental metabolic pathway that converts glucose into pyruvate. It provides energy in the form of ATP and NADH for cellular activities. Glycol cleavage is a key step where a 6-carbon sugar is split into 3 carbon molecules. Overview of glycolysis. Glycolysis occurs in the cytoplasm of cells, occur all aerobic and anaerobic organisms. It consists of 10 enzymatic reactions that transform glucose into pyruvate. The pathway is crucial for energy production and serves as a precursor for other metabolic pathways. The glycolytic pathway key steps. The pathway begins with the phosphorylation of glucose to glucose 6-phosphate. It proceeds through a series of reactions producing intermediate like fructose 1,6-phosphate. The final step generates ATP and NADH, culminating in the formation of pyruvate. Glycolysis in detail. Energy investment phase. In the first phase, 2 ATP molecules are consumed to prepare glucose for cleavage. Glucose is converted into 2,3-carbon sugars, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This phase sets the stage for energy payoff in subsequent steps. Glycol cleavage, the critical step. Glycol cleavage occurs at the enzyme aldolase, which splits fructose 1,6-bisphosphate. The cleavage produces 2 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. These molecules are interconvertible, allowing the pathway to proceed efficiently. The role of aldolase in glycol cleavage. Aldolase catalyzes this reversible cleavage of fructose 1,6-bisphosphate into 2 triose phosphates. It uses a shift-based mechanism involving lysine residues in its active site. This step is essential for the continuation of glycolysis and energy extraction. Conversion of dihydroxyacetone phosphate. Dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate by triose phosphate isomerase. This ensures that both molecules can proceed through a subsequent step in glycolysis. The equilibrium favors glyceraldehyde 3-phosphate, maximizing energy yield. Glycolysis energy payoff phase. Glyceraldehyde 3-phosphate is oxidized and phosphorylated to 1,3-bisphosphoglycerate. This phase generates ATP and NADH, providing energy for the cell. The pathway continues with substrate-level phosphorylation to produce ATP. Significance of glycolysis and cleavage. Glycolysis is vital for cellular energy, especially under anaerobic conditions. It provides intermediates for biosynthetic pathways such as amino acids and lipids. Understanding glycolytic regulation helps in confronting metabolic disease like cancer. Regulating of glycolysis. Key enzymes like hexokinase, phosphofructokinase, and pyruvate kinase are tightly regulated. Allostatic effectors such as ATP, ADP, and straight molecule enzyme activities. Regulation ensures balance between energy production and consumption. Clinical and biotoxicological implications. Abnormal glycolysis associated with cancer cell proliferations known as the Warburg effect Enzyme in glycolysis are target for drug development in the metabolic disorders. Engineering glycolytic pathway has applications in biotechnology and biofuel production. Summary and conclusion of glycolysis and glycol cleavage. Glycolysis involves a series of reactions including critical cleavage step to generate energy. Glycol cleavage splits fructose 1,6-bisphosphate into 2-triose-phosphate, fueling the pathway. Overall, glycolysis is essential for energy metabolism and cellular function in all organisms. This is all about my presentation. Thank you so much.